So in 2019, TGEN, located in Arizona, started a dedicated OCNDS program. And so we are pleased to welcome Dr. Matt Huentelman to learn more about their program.
Thanks a lot. Appreciate the invitation to be here and really value everybody's really teaming up together and trying to understand OCNDS. I'm going to tell you a little bit about what we're doing in Petri dishes in the lab. But first, I am going to repeat a few things because it's really important to go into those details a little bit to make sure that we're all on the same page. And that's the most important thing, is to making sure that the families here understand what us scientists are talking about when we say strange words like RNA. So I'll go into that in a little bit. This is my team. I'm a neuroscientist and I'm from TGEN in Phoenix, Arizona. We're a nonprofit academic research institute. And I'm a geneticist. We study genetics and how it influences human health and disease. We focus on rare diseases, but the other half of my lab focuses on age-related diseases like Alzheimer's. And I just want to recognize the two scientists who are really important for this OCNDS program, Aya Kamzina and Lee Nicholson. Aya is here today in the audience, so you might see her and say hello during the break. Lee went back to her home in Australia a few months ago. We were sad to see her go, but Aya and I wanted to recognize Lee's real contribution, both in her thoughts, but also with her hands for the work that I'll tell you about today. And lastly, we want to thank you guys. It has been really an incredible ride. And just so much fun to work with the foundation and the patients and their families. It's just really from the bottom of our heart, we thank you for supporting this, and also the researchers. It's been just a wonderful collaborative community, and you don't get that all the time in the stuff we do. So it's been really wonderful. Okay, let's do Genomics 101. Maybe this should have been the first talk. Um, all right, so our bodies are made of organs, our organs are made of cells. And different organs have different complexities, but they tend to be made up of really specialized cells, and they can be hundreds of them, hundreds of different types of cells that are doing all these different functions. The strange thing is, they have the exact same DNA inside each one of those cells. So the instruction manual, that's how I like to think about DNA. Some of you can think about it like it's the blueprint, the book of life, however you want to think about DNA. Those are some ways you can conceptualize it. It's the same in every single cell in our body. Couple exceptions, though. There's always exceptions in science, right? Your sperm cells and your egg cells, they only have half the DNA. That's a good thing, because when they get together, then you got a complete set. So your sperm and egg have half your DNA sequence. And then when it comes to cancer, oftentimes you have duplications or deletions. So it's not all your complete instruction manual. There can be a bunch of pages that might be copied 20 times in a particular cancer, for example. But in general, for what I want you guys to care about today, it's really quite amazing that we have this incredible collection of different organs inside of our body that do different things, but they all have the same instruction manual, your DNA. So how does that work? Here's how I like to explain it. And if you work in construction and made your money in construction, or feed your family by doing construction, I apologize. I'm going to get this entirely wrong. But I'm a scientist. But this is how I like you guys to think about how this works inside of our cells. Okay, so now we're inside the cells, and we're going to talk about DNA, RNA, and proteins. And we're also going to talk about the environment. So you've heard all of us scientists say these words, right? What do we mean when we say environment? What do we mean when we say RNA? Let me try to explain it to you. Just give me a few minutes. Okay, so if you imagine a construction site like I do, it starts with the blueprint. That's essentially the plan for the building. And it lives in one spot on the construction site, right? The blueprints don't get passed around. They sit in this beautiful trailer. It's air conditioned, super important in Arizona, and they're laid out on a big table. That's how I imagine how it works. That is like your DNA sitting inside the nucleus of your cells because it's important and it's protected inside the nucleus of your cells. So that's the blueprint on the construction site. Now, each morning, something has to happen, right? Things have to get done when they're building the building. And the way I imagine that happens is there's a bunch of project managers. Some of them are doing electrical work, some of them are doing plumbing. But every day they have to come in and they have to check that blueprint and figure out what they're going to tell their teams to do that day. Those project managers, they go and they look at the plan and then they take it out into the job site. That's kind of like RNA, okay? RNA is like a sister chemical to DNA, and it goes from the nucleus into the rest of the cell, and it essentially carries instructions, okay? That's why I say that RNA is like a project manager, carries the instructions out into the cell. When RNA gets out there into the cell, it's formed into a chemical or a collection of chemicals that we all know as, and we call them proteins. Okay, so RNA encodes the instructions to build these proteins, and then proteins do stuff in our bodies. Okay, they metabolize that caffeine, unfortunately, so you have to go for another cup of coffee. They digest your food. They do stuff. Okay, now of course, to my scientist friends, I'm simplifying things here, but this is the general concept. On the job site, the proteins are the actual workers on the ground who are laying the electrical pipe, the electrical lines, laying the plumbing pipe, doing the construction work. Okay, they're the ones building stuff at the construction site. So that's DNA, RNA, and protein. The last piece of the equation for purposes of today is the environment. Who'd you purchase your cement from to pour your concrete? Who'd you buy your electrical wire from? Was it a good supplier? Was it a poor supplier? That can harm your building if you have the perfect plan, the perfect blueprint, but you purchased some pretty poor cement, then you can pour a bad foundation and have a not properly built building, right? So the environment is a really important part of this equation. So that's DNA, RNA, protein, and environment. We good? All right. So now you have a little reference point when one of us gets up on stage and we say RNA or protein. That's the basics of it. So how does this look in the context of OCNDS? We can go there right now and talk about it. So remember, the change that happens with OCNDS is a change in the genetic sequence in that DNA for the CSNK2A1 gene, right? That's a big mouthful, but now you guys understand all of that now. This is a change in the blueprint specifically at that CSNK2A1 gene, okay? Change in the DNA. And now you can understand that if those project managers, the RNA in your cell, is produced from that DNA, it can take the wrong plan out to the rest of the cell because of that misspelling, that mutation, that change in the DNA. So now you have a bad plan and you end up having a problem with building the proper proteins to do that work in the body. Okay? And so I've listed a bunch of gobbledygook that the proteins, the things that are affected by the CSNK2A1 genetic mutation, cell proliferation, DNA repair, RNA metabolism, bunch of science gobbledygook. To you guys, it means sleep problems, behavior problems, all the things that we talked about already today. So because of that change in the DNA, there were some bad instructions, and the protein that was made can't do its proper job for each of those things there. And then we see it as what you guys deal with every day as parents. How's the environment play a role? Well, we know, and even Dr. Chow talked about how some supplements were hits on his screen. Sometimes you can have effects by changing the environment. And in this case, what I want you to think about are like dietary changes. Some of you may have tried these dietary changes to control epilepsy, for example, right? So that's a way where you're not really trying to alter the protein, the DNA, or the RNA, but you're kind of changing that environmental side to see if you can have an impact, a favorable impact. All right, makes sense? All right. I spent a lot of time depending on what that timer is telling me. That's counting down to 0, right? Then I stop. Okay. I also want to talk about modeling the brain. And we heard a lot of people today talk about models. What does that mean to a scientist? Well, if we didn't have to do a model, we would study the patients. So that's why we try to study the patients as much as we can, but sometimes we need to do models. I hope you can probably appreciate that modeling the brain itself is really, really challenging. Um, we don't even know for sure how many different cell types are in our brain, but it's probably close to hundreds to even thousands of different cell types in our brain, which is really pretty amazing because when I learned about the brain in school, I just thought it was, this blob that was pretty, similar all across the whole brain. The other, and I'm showing you some pictures of some cell types. There's neurons and astrocytes because they look like stars. And there's these other crazy looking cells called microglia. They take care of, they're sort of the immune system of your brain. The center picture there, that's not the real color of your brain. That's all colored by these areas that scientists call Brodmann areas. They were defined by a researcher, last name of Brodmann. And there's about 50 of these. And the reason I'm showing you this is just to remind you that actually these represent specializations within your brain. So there is a specific part of your brain that's important for sleep, important for movement, important for smell, and things like that. And those have been defined scientifically and experimentally. So your brain doesn't participate in every single thing you do. It actually has specialized regions, which actually makes it even harder to model if we can't study the whole brain. If we have to study it in a dish, it becomes really hard to model. But you've heard about model organisms already: mice, rats, flies, worms, fish, and tadpoles. I think these are the top 5. Tadpoles is number 6 on the list. So I'll let it squeak in today in honor of Unravel. These are all really powerful for all the reasons you heard about today, but they're called models because it's not actually able to give information about the human brain, but really, really powerful. I'm going to tell you about a different model today, just briefly. And this is a model that we call in vitro modeling, literally translates as in glass. Because back in the old days, Petri dishes that we would grow these special cells in were made of glass. So here's what we do. We typically start with a skin biopsy for this work, which is a very small punch of skin. And you can grow these cells here that I'm showing you. They're called fibroblasts. They are the weeds of the human body. They grow like crazy. Okay. And using a special molecular trick, we can turn them into these cells, which are in the middle there. And it's the worst series of words we're going to say today out loud. Okay. Human induced pluripotent stem cells. Okay. Lots of words. All you need to know is that through this molecular trick, we can turn those fibroblasts into these very interesting cells that can actually then form all the cells in our body. So if you can take those cells and convince them to form brain cells or heart cells or muscle cells. That sounds amazing, right? And it is amazing, and it was awarded the Nobel Prize. This technique alone was awarded the Nobel Prize very recently. So that ability to turn these cells like fibroblasts into these pluripotent stem cells is really very powerful. In this experiment, we're going to turn them into brain cells, both neurons, give you a little cartoon there, and astrocytes, the cells that look like stars. And we're going to study them. Because what we're trying to understand are the differences between the genetics and the phenotype. So we've said phenotype a lot today too. Phenotype is the thing that you can see, how things look, how things behave. And we all know that there seem to be some differences between the genetics and what we see in the patients. So this is complicated. It's really not important. The point is we're covering 4 different very common or, starting with the most common genetic changes that cause OCNDS, and we're going to study those in a dish in our laboratory. And we're going to study both the neurons and the astrocytes as our model cells. This is important to do because it lets us look at the differences between these 4 different genetic changes and also across these 2 different really important brain cell types. We're also going to study a bit of what happens across time, so how these molecules change as we're growing these cells in the little Petri dishes. You don't even need to pay attention to this slide. It's just showing you that, hey, we're doing a lot of stuff. We're doing a lot of different science assays. So we're trying to do a lot of the obvious stuff right here first and try to really generate some data that all of us can look at together. So here are some cool pictures because these are actually the cells that we're working with. So this project just started. I don't have a whole lot of data to show you other than some of the pictures. But we wanted you to be aware of this because this is being supported by the foundation funds. And the idea is to help scientists in this room who want to have access to these data have access to it as quickly as we have access to it ourselves and generate samples that they can use. Regardless, here's some pictures of these cells already growing in these little Petri dishes. And we're already starting to turn them into brain cells. So this is the first step in turning them into both astrocytes and the neurons. And the reason I'm showing you this slide is kind of, you guys asked, how long does it take to do the fly stuff? How long does it take to do the tadpole stuff? This also takes several months to actually go from start to finish. And I wanted to talk about some of the challenges and the pitfalls too. Growing these cells is really very individualized. So what's neat about this, but also what's frustrating about this is where science becomes art. Most scientists would prefer science would remain science, something that's predictable and reproducible. But when it comes to these cell lines, they are very individualized, and sometimes they can behave different than each other. So one needs a little more attention, or one needs a different type of feeding schedule when it comes to growing them. So it's actually a really a significant challenge here. The other thing is that when we do this and we generate the items that we're going to share with the scientific community, we can't do everything, and we're always going to run out of stuff to share. This is a finite resource, but that's okay. We can start it again, but it'll still take months to catch back up. So I'm just trying to help you guys understand some of the challenges with science that prevent us from moving as quickly as we can. These are finite resources. But nonetheless, we're already starting to see differences. So what I'm showing you here is a cell line that we developed. So these are now these are fake colors, okay? So the other ones were actual pictures through the microscope. These are colored based on things that we're trying to investigate. So the green color represents neurons in this sample, and the blue color is a stain for DNA. So you can see the blue looks like little circles, which represents the nucleus in the cell, and the green is showing you where the neurons are. And if you just look on the right side, the two figures on the right side, on top is what a wild type, normal, healthy uh, batch of neurons look like in this particular example. And on the bottom is the CSNK2A1 mutation, the L198A. Okay, that's just a particular genetic change in the CSNK2A1 cells. And hopefully, even just by you guys staring at it, you can see that the green neurons on top have a lot of really long, spindly processes. They look like how you would draw a brain cell, right? And then the green cells on the bottom, they just look different. They look like they have shorter processes. They're not stretching their arms out so much. So visibly, we can see a different change here, a difference between these two different cell lines. That could represent something interesting to learn from the particular OCNDS genetic change in the cells on the bottom. So why does that matter? It's helpful for us to learn new things because it might help us prioritize drugs or prioritize new approaches to find new medicines. Maybe this is another thing that Dr. Novak and Dr. Chow want to add to their next screens. They might want to say, well, the second step is to see if it works in these type of, changes these type of things in cells and culture. They might want to do that before or after the tadpoles or before and after the new fly test, something like that. So it can provide additional information, and it can also play a role in the drug screens. Okay, so just to summarize, we're performing this study on 4 different known OCNDS causing genetic changes, DNA changes in patient-derived cells. So these are from the graciousness of your donations into this resource. These are derived from OCNDS patients. We're going to collect and study RNA, protein, and the cells themselves. Now you guys know what RNA and protein is, right? We're going to study that, and we're going to share that with other scientists so they can apply their own ideas and their own tools to look at those changes. And one of the big things here is to understand those differences between the genetics and what we can see in the dish, the genotype and the phenotype relationship, because we know that's a big struggle with how we manage and try to understand OCNDS clinically. And lastly, I already showed you that we're seeing something very interesting. We're already seeing a difference. And so there's likely a whole lot for us to learn as we expand the study. And there is my contact information. Happy to answer any questions.